Imagine standing in front of a honeybee hive. It's just this scene of incredible activity, thousands of bees constantly moving. They seem independent, right? Each with their own job.
Mm-hmm.
Is that all you're seeing, though? Just a huge crowd of busy individuals?
Yeah.
Or is there something else, I don't know, much more fundamental going on?
Yeah, that question, it really gets to the heart of it, doesn't it? It's exactly what we're diving into today.
Yeah.
We're exploring one of the most powerful and, frankly, pretty debated ideas in biology, the concept of the honeybee colony as a superorganism.
A superorganism. And you, our listeners, sent us a really fascinating stack of material, articles, research notes that really digs into this complex notion.
Yeah, absolutely. So our mission today is to navigate these sources together. We want to understand what thinking about a bee colony as a superorganism actually means, in scientific terms.
Right.
And why it sparks such debate among researchers and what it tells us about how these colonies, well, how they really work. We're aiming to pull out the essential insights without getting too bogged down in the academic jargon.
And I should say, the takeaways are pretty surprising. We're going to see how bee colonies function in ways that are remarkably similar to individual animal bodies. And crucially, why studying bees just on their own in isolation can actually lead us down some totally misleading paths.
Exactly.
All right, so let's just jump right in then. This superorganism idea.
Yeah.
It sounds a little bit sci-fi maybe. What does it actually mean?
Well, at its core, the concept proposes that a highly social insect colony, like a honeybee hive or certain ants or wasps, isn't just a group of individuals living together.
Okay.
It can be viewed as a single, integrated, functional unit. Think of it operating much like, say, a multicellular organism like your own body.
So instead of a body made of cells, it's kind of body made of individual insects.
That's a thick analogy, yeah.
Yeah.
But as our sources make really clear, it's not universally accepted. It's actually a pretty contentious issue in social insect biology. Many researchers feel it's absolutely necessary for understanding the colony's complex organization. Others, though, including some philosophers, worry it might be more of a catchy label, but potentially, a distraction.
Okay.
The sources definitely lay out the arguments on both sides of that debate.
Okay, that makes sense that there's disagreement. I mean, my first thought hearing superorganism was, isn't that just a metaphor?
Yeah.
Like saying a company is like a living organism.
That's exactly the criticism, and it's addressed directly in the sources. They make a really critical distinction here.
Yeah.
They argue the similarity between a colony and a multicellular organism isn't just poetic. It's not a simple metaphor. It's based on functional similarity. And that similarity is driven by something called convergence.
Convergence. Okay, what's that?
Right. So in natural selection, convergence is when completely unrelated lineages independently evolve very similar solutions to the same environmental problem.
Oh, okay.
The classic example is the body shape of dolphins and sharks. One's a mammal, one's a fish, completely different origins, but selection for moving efficiently through water led to these remarkably similar, streamlined forms.
Right, like nature finding the same good design multiple times over.
Precisely. Another powerful example the sources use is wing design in birds and bats. Very different origins, obviously, but the problem's the same. How do you achieve flight? How do you generate lift and thrust? Natural selection converged on the wing structure.
Okay.
And the sources even point out that researchers use surprisingly similar equations, just kind of reparameterized to study the aerodynamics of both bird wings and bat wings.
No kidding.
Yeah, because the underlying physics of generating lift with that shape are analogous.
Right.
Even, fixed wing aircraft share some principles. You also see it in things like farming symbioses, ants and termites independently figured out how to farm fungi.
Wow, that's a cool detail. So applying that idea back to bees and organisms.
The argument from this perspective is that the deep functional similarity between a social insect colony and a multicellular body isn't just coincidence or some poetic comparison.
Right.
It's because natural selection is tackling essentially the same core problem in both cases. How do you maximize the reproductive success of a collective unit made up of many interacting parts?
Whether those parts are cells or individual bees.
Exactly. And the organizational structures and processes that selection favors to solve this problem, they end up being functionally analogous. Similar solutions to similar problems.
Okay, I think I'm getting it. So when researchers use terms like anatomy or physiology for a whole colony, they're not just being flowery or cute.
Not according to these sources, no. They argue that using that kind of common terminology isn't just a linguistic flourish. It reflects these deeply similar, selection-driven ways that both organisms and superorganisms are organized and function. The terms are appropriate because of the analogous adaptations that result from this convergence.
All right, so if we're thinking of it like an organism, what are its parts? What makes up the anatomy of this superorganism?
The absolute central adaptation that the sources identify, and it's critical for both multicellular organisms and these social insect superorganisms, is division of labor. DOL.
Division of labor.
Yeah.
Like my body having different organs for different jobs. Heart, lungs, that kind of thing.
Precisely that analogy. In our bodies, you have specialized organs. In a social insect colony, DOL shows up as different individuals specializing in different tasks.
Okay.
Now, this can be through physical castes, where individuals are morphologically different, think queen versus workers.
Right.
Or it can be through temporal castes, where workers specialize in different tasks as they age, like young bees do one thing, older bees do another.
And why evolve this? What's the advantage?
Greater productivity. It comes down to specialization effects. When an individual can just focus on doing a limited set of tasks, they generally become much better and more efficient at them.
Makes sense.
The source gives a simple but I think powerful honeybee example. Imagine a forager finds a really rich patch of nectar, but it's only going to be around for a short time.
Okay.
If she specializes purely on collecting that nectar, she can make trip after trip really quickly. But if she also had to spend time back at the hive, processing that nectar into honey herself, well, she'd make far fewer trips out to the source.
Right, she'd be wasting valuable foraging time.
Exactly. Specialization increases her rate of resource acquisition for the whole colony.
Okay, I see. So you've got these specialized parts through division of labor, but just having parts doesn't automatically mean they work together effectively. How do all these different bees doing different jobs actually coordinate? How is the physiology managed across the entire colony?
Right, and that's where this idea of social physiology comes in. And it's a term that actually grew out of decades of, observing and experimenting with colonies. It wasn't just a theoretical idea pulled out of thin air.
Yeah.
It's about the mechanisms the colony uses to coordinate activity at the group level.
That sounds incredibly complicated, like trying to manage a massive, constantly shifting team project or something.
It absolutely is. The sources use a human analogy that helps illustrate it. Imagine a construction team building a brick wall.
Okay.
They use DOL, right? You've got bricklayers, mortar mixers, laborers carrying bricks. That definitely increases output compared to everyone trying to do everything.
Sure.
But if there's no coordination, say you have too many bricklayers just standing around waiting because there aren't enough laborers bringing bricks and mortar, then your overall efficiency just plummets.
Yeah, bottlenecks.
Exactly. You need communication and coordination to keep the optimal ratios balanced, and those optimal ratios might even change throughout the day depending on conditions.
Okay, that makes perfect sense. So what are some actual examples of this social physiology in action? What do the sources highlight?
Well, Robert Jean's work on social wasps is a classic study they mention. He looked at different wasp species, comparing small colonies like Polistes paper wasps to much larger swarm-founding wasps like Polybia occidentalis.
Okay, big difference in size there.
Huge difference. And he noticed these large Polybia colonies use something that looks a lot like assembly lines for tasks like nest building.
An assembly line for building a wasp nest. How does that work?
Workers specialize. You've got some that just collect wood pulp, others just collect water, and then others are the masons who actually mix it and build the carton wall.
Wow.
And the sources confirm there are benefits to this, like spiralized wasps can carry heavier loads of their specific material. But UT, and this is the really crucial part, Devine found that in small colonies, this assembly line approach actually doesn't work very well.
Why not?
If you have fewer than about, say, 50 workers available for one particular task, the costs of queuing delays, a builder waiting for pulp, a water collector waiting to unload her water, those delays actually outweigh the benefits of specialization.
Okay. Not enough players to keep the chain moving smoothly. Lots of waiting around.
Precisely. Small colonies just can't afford that waiting time. Large colonies, however, can maintain these assembly lines effectively.
And that makes them more efficient.
Yes, and the sources note this leads to a non-linear increase in per capita work output as colony size increases, meaning bigger colonies aren't just proportionally more productive, they're more productive per individual worker when they can use this kind of coordinated system.
Interesting.
What's fascinating, though, is the sources mention Gene wasn't able to figure out the exact mechanism. How do the wasps coordinate allocating the right number of workers to these different specialized groups? He saw the pattern, the assembly line, but the how was elusive.
Okay. So that's wasps. Where do honeybees fit in? Do we know more about their coordination mechanisms?
Yes, that's where Tom Seeley's work comes in prominently, particularly from his book, The Wisdom of the Hive, which is referenced in the sources. He provides more insight into those specific coordination mechanisms.
Okay.
He's looked extensively at how honeybee foragers coordinate with the middle-aged bees, the MABs, who act as the nectar receivers, processors, and the ones who store it.
Right, the crucial links in the honey-making chain inside the hive.
Exactly. And it's critical because if there aren't enough bees receiving nectar, or if the storage space runs out, the whole foraging operation can get jammed up or even shut down completely.
Okay.
Seeley did experiments confirming this. If you remove the nectar receivers, foraging just stops. But the foragers didn't just sit there idly.
What did they do instead?
They started generating signals to try and alleviate the bottleneck. And one key signal, detailed really well in the source material, is the tremble dance.
The tremble dance. Okay, tell me about that.
When a forager comes back loaded with nectar, but she has to wait a really long time to find a receiver bee to unload it to.
She gets impatient.
You could say that. Instead of just waiting near the hive entrance, she'll start walking through the nest, performing this distinct shaking or trembling dance. She's broadcasting a message throughout the hive.
A tremble dance. What's the message? Hey, I need someone to take this nectar.
That's Seeley's hypothesis, yeah, and it's backed by the evidence in the sources. The dance signals a need for more nectar receivers. And sure enough, soon after a lot of tremble dancing happens in the hive, more workers shift tasks and become receivers. The bottleneck clears and foraging can ramp back up.
Wow.
So the idea is foragers follow a pretty simple rule. If the wait time to unload is short, great, keep foraging. Maybe even do a waggle dance to recruit more foragers to that good source.
Right.
But if the wait is long, stop foraging for a bit and perform the tremble dance instead. This prompts other bees to switch jobs and become receivers.
So it's a feedback loop controlling the workforce allocation.
Exactly. Now, the sources do caution the tremble dance might have other functions too. It's complex. But this role in coordinating nectar processing is a really key finding.
Okay, this is where it really starts to shift my thinking. How does actually viewing the colony as a superorganism fundamentally change how researchers should approach studying these bees?
Well, according to the sources, taking this superorganism perspective is absolutely crucial. It helps avoid some really fundamental research errors. They highlight two major pitfalls in particular.
Okay, lay them on me. What are the pitfalls?
The first one they talk about is what they liken to an in vitro versus in vivo problem. They draw a direct analogy from molecular biology.
Right.
In molecular biology, if you study cells in a petri dish that's in vitro, they often behave very differently than they do when they're part of a whole living organism in vivo, where they're connected to all the complex signaling networks and environmental cues inside the body.
Right, context matters.
Hugely. And the sources argue a single bee worker is exactly the same. She's not really an independent agent acting alone. She's deeply embedded in this dense, complex social network within the hive, and that network dictates much of her physiology and behavior.
So studying a single worker bee in a lab cage, that's like studying a cell in a petri dish. It's missing the bigger picture.
That's precisely the argument. It's stripped of all those essential colony-level inputs that are normally regulating it. And the source provides a really eye-opening example with studies on kin recognition.
Kin recognition, like bees recognizing their sisters.
Exactly. Some early research, often using bees that were reared in isolation or small artificial groups, showed that workers seem to have a preference for associating with their close kin, their full sisters, over their half-sisters, for example.
Okay, and that suggested?
It suggested there might be potential for internal conflict within the colony, maybe workers trying to favor their own relatives, which could undermine colony cohesion, nepotism, basically.
That sounds like it could be a big deal for how a colony functions.
It did seem like a big deal. But here's the twist. When those experiments were repeated more carefully, with bees reared in larger groups, or actually within a functioning nest, the in vivo condition, essentially.
Let me guess, the preference disappeared.
Completely disappeared. Vanished. The explanation offered is that a gestalt mechanism operates within the nest.
Gestalt mechanism.
Yeah, basically, the colony develops shared chemical cues or labels, a kind of colony odor profile that creates a single unified colony identity. Everyone recognizes us, the colony members, rather than focusing on individual kin differences among the workers.
So the isolation created an artificial situation.
Exactly. The result showing kin preference and isolation was likely an experimental artifact. It was caused by removing the bees from their natural social context, from that unifying colony environment.
Wow. So what looked like a fundamental individual behavior in the lab wasn't actually happening in the real hive.
Precisely. And the crucial point the sources emphasize here is that results obtained from artificial settings, like small cage studies, lab bioassays, should always be treated as preliminary hypotheses.
Until they're checked in a real colony.
Until they can be verified in whole functioning colonies. They note that this verify in vivo standard is pretty much mandatory in molecular biology. You wouldn't publish a major finding based only on tissue culture results. But this perspective, this standard, isn't as deeply ingrained or consistently applied yet in social insect biology, and that can lead to potential misinterpretations of bee behavior and physiology.
That's a really powerful point for anyone doing research in this area.
Mm-hmm.
Okay, what's the second big error the superorganism view helps avoid?
That one's related to what they call distributed physiology. It's the idea that in a superorganism, many processes that in a solitary insect would be controlled entirely within that single individual, while they get shifted and distributed across the entire colonial level.
Distributed physiology, can you give me an example of that?
Sure. Think about something really basic, like hunger. A solitary insect, say a fly, gets hungry. Its internal state changes, blood sugar drops, whatever, and that directly triggers it to go out and forage for food.
Makes sense. Individual need drives individual action.
Right. But honeybees, they're different. Individual worker bees don't typically experience that same kind of individual hunger driving them to forage. Why? Because the colony usually maintains massive food stores.
The colony larder is full.
Often, yes. So honeybees forage not usually because they are individually hungry, but because nectar is available, or because the colony as a whole needs pollen for the brood, for example. It's a colony-level need or opportunity that drives the behavior, not usually an individual's internal hunger state.
Okay, so the decision point, or the trigger, it's moved up from the individual to the colony level.
Exactly. Or consider ovary development. In a solitary female insect, ovary development is regulated by hormonal signals from her own brain and endocrine system. Pretty self-contained.
Right.
Honeybee workers still have these hormonal systems, of course, but the forces point out they are operating downstream of signals coming from outside the individual specifically, signals from the queen and from the developing brood.
So the queen and brood are influencing the worker's internal state.
Profoundly. The complex network that controls whether a worker's ovaries develop or stay suppressed is much larger than just her own body. It's distributed across the queen, the brood, and the interacting workers themselves through chemical signals, mainly pheromones.
So the control isn't just inside one body, it's spread out across the whole colony.
That's exactly right. Many regulatory processes that govern development and behavior processes that rely on endocrine signaling within an individual in solitary insects become dependent on exocrine signaling. That's chemical communication between individuals in these highly social colonies.
Okay.
This means the control network is physically larger, spread out across multiple individuals and castes. And consequently, the gene networks underlying these social traits are also likely larger and more complex than in their solitary relatives.
Which means you can't just study them the same way.
It means you simply can't take research methods or assumptions designed for simpler solitary systems, like, say, studying gene function in a fruit fly, Drosophila, and apply them directly to a complex social insect like a honeybee without accounting for this fundamentally different distributed control system. You'll miss crucial parts of the picture. So ultimately, our sources describe this as highlighting two fundamental worldviews, or maybe two different lenses you can use when you're studying insect societies.
Two different ways of seeing the same thing, like the hive.
Essentially, yes. First, there's what they call the society view.
Okay.
This perspective sees colonies as complex animal societies, maybe like a wolf pack or a lion pride, but, more intricate. Individuals are seen primarily as independent agents, maybe cooperating, maybe competing. And their behavior is understood mostly in terms of individual interests, even when they're interacting. Communication in this view might involve elements of conflict or manipulation, so ensuring honest signals between potentially competing individuals becomes really important.
Okay, so focusing on the individual within the group.
Right. And the sources suggest this view is actually quite appropriate and useful for understanding some species, like maybe Polistes paper wasps, where colonies are smaller and there might be more direct reproductive competition among females.
Okay, that makes sense. Independent actors in a complex social structure. And the other view?
That's the organism view, the superorganism perspective we've been focusing on. This view sees the colony itself as the primary agent, the single functional unit with many parts.
The individuals are like parts of the machine.
Or parts of the body, yeah. Individuals are viewed more like components, like cogs or organs, and their behavior is best understood and predicted in relation to achieving colony-level goals, survival, growth, reproduction of the colony.
And communication in this view
is generally seen as honest and cooperative. Like the honeybee dance language system, it's seen as a way for parts of the organism to efficiently share information for the benefit of the whole. There's less focus on potential conflict between individuals because their fates are so tightly linked to the colony's success.
Right.
So this organism view naturally directs research towards understanding the connections between individuals, the flow of information, the signaling mechanisms like pheromones or dances that coordinate the whole unit.
And it forces you to see distributed processes.
Exactly. It forces researchers to confront the fact that processes happening within a single body in other animals, like regulating hunger response or ovary development, are often distributed across many interacting bodies in a social insect society, like a honeybee hive. It's a fundamentally different way of being organized.
Wow. Okay. So stepping back, what does this all really mean for us thinking about this? We've taken a pretty deep dive, thanks to these sources, into this idea of the honeybee colony as a superorganism.
And we've seen how that concept isn't just some loose metaphor. It's argued quite strongly in these materials to be based on functional convergence. You know, unrelated biological systems arriving at similar effective solutions through evolution.
Right, solving similar problems of organization and reproduction.
Exactly. We've explored the key elements of that convergence, the highly specialized division of labor, which acts kind of like social anatomy.
The different parts doing different jobs.
And the complex social physiology, the mechanisms that actually coordinate all those individual parts. We looked at fascinating examples like the tremble dance, helping regulate resource flow in the hive.
And, crucially, we've learned why adopting this superorganism perspective seems vital for doing sound research.
Yeah.
It helps scientists avoid potentially significant errors.
Yeah, like misinterpreting behaviors seen only in isolated lab settings that in vitro trap.
Right. And understanding that many fundamental biological processes, like regulation and control, aren't just confined to a single individual bee, but are distributed across the entire colonial network. It really changes how you have to think about what's going on.
It really does. It changes what you see when you look at that bustling hive. You start seeing the connections, the coordination, the colony-level patterns emerging from all that individual activity.
And this isn't just limited to bees, is it? I mean, this deep dive, it gives you, the listener, a new kind of lens for thinking about complex systems in general.
Absolutely. It's about how specialization and coordination and this seemingly unified function can emerge at different levels of organization, built up from simpler components interacting in sophisticated ways.
Which kind of leaves you with a thought to carry forward, doesn't it? Something from the sources to mull over. Consider how many processes that you might instinctively assume are controlled and contained within a single unit, whether that's an individual person, a single computer, maybe a single department in a company.
Right.
How many of those could actually, in different contexts or systems, be distributed across a much larger interacting network of parts? What other organisms, hidden in plain sight, might be operating all around us, just waiting for us to recognize them as a functional whole?
That's a great thought to end on. Thanks for joining us on this deep dive.